Welcome to Balliol.
So the subjects for everybody are issues like neo-Darwinism and what the sources of evolutionary novelty are, group selection, groups, populations, communities, ecosystems as units of selection, thought styles of neo-Darwinism and other things, and I just listed that. So that all of those subjects are open. Any aspect of evolution is open for discussion when we get started. Okay, Dennis, now. Tell him. Yes, that's fine, thank you.
I'm going to stand up most of the time because my job is largely to control you all. As Lynn Margulis has said, we really do want this to be interactive discussion. Well, with no more ado, I think we ask, to get it in the order as you said, Lynn, we ask Stephen Bell to give us his 5-minute view on the origin of life.
So I'm actually a biochemist. My lab's studying biochemistry, processes such as DNA replication, gene expression, cell division. And in doing so, we're studying these peculiar little organisms, the archaea, which are simple prokaryotic organisms, devoid of any interesting features really whatsoever. You can see they're simply a bag of protein and DNA. And they certainly have nothing like the complexity of present-day eukaryotic organisms with their multitude of subcellular membrane-bound organelles and complex trafficking systems. But the biochemistry that we've done, I think, is beginning to help shed some light on the evolution of these vastly more complex eukaryotic organisms. And that's what I'd like to talk to you about briefly today and just highlight a few of the questions that interest us. So thanks to Carl Woese, we now know that there are 3 principal domains of life. There's 2 prokaryotic domains, organisms that lack nuclei and are devoid of subcellular structure, as I showed you. And that's the bacteria and the archaea. And then, of course, we have the eukaryotic organisms. And what's been central to our studies is the observation that the archaea appear to share a period of common evolutionary history with the last ancestor of eukaryotes. And it's during this period of shared common evolutionary history, which has been distinct from the bacteria, that certain core machineries have evolved, and we've been able to exploit these in simplistic studies of biochemical function. Within the archaea themselves, there are 2 principal phyla have emerged, the Krenarchaea and the Euryarchaea, and I'll come to this in a second or two. So the first hint really that there's this special relationship between eukaryotes and archaea came with studies of the gene expression apparatus, the transcription machinery. And recently we've established the crystal structure of the archaeal RNA polymerase, which highlights its complex nature and its similarity to the eukaryotic enzyme, and shows how much more complex and how distantly related it is to the bacterial enzyme. Similarly, when we look at the process of DNA replication, a process that's central to the propagation of all life on the planet, it's a complex multi-stage process. The bacterial proteins that carry out all these various transactions on DNA leading to its duplication are very well understood. But through an amazing feature of evolution, we have an entirely distinct machinery for DNA replication that's found in present-day archaea and eukaryotes. Again, from my lab's point of view, this is really useful. The archaeal machinery is a simplified version of the eukaryotic. It's a useful model system. But how did this happen? How do we have 2 entirely distinct mechanisms driving DNA replication? 2 entirely distinct sets of proteins. And finally, I mentioned already, the eukaryotes have this incredibly complex endomembrane systems with shuttling of vesicles backwards and forwards between these membrane-bound organelles. How did this evolve? How did this come about? Well, it turns out that one of the principal players in shuttling between these membrane-bound organelles is a eukaryotic apparatus called the escort machinery. And very recently, my lab's discovered that archaea actually possess this escort machinery. But these things don't have subcellular organelles. What instead it turns out the archaeal escort machinery is doing is actually carrying out cell division. So we have an entirely distinct process of binary fission occurring in the archaea that is, if you like, the ancestor of the eukaryotic membrane shuttling systems present in these complex organisms. So if we look at the archaea, we see this whole list of eukaryotic-like features. We have, just to mention some of the ones I've alluded to today, multiple origins of replication driving DNA replication, various proteins that are much more closely related to eukaryotic than bacterial. We have the escort machinery. We acetylate our chromatin proteins, the way we package DNA. These are all features that were thought to be typical of eukaryotes. But what we've discovered is that these features are present in the Krenarchaea, but thus far there is absolutely no evidence whatsoever for them in the Euryarchaea. But they're also present in Eukarya. So this is a slight problem in the classic Woosian tree of life. Have all these features evolved independently twice, once in the Krenarchaeal lineage, once in the Eukaryoal lineage? Or is the topography of the tree of life, as proposed by Woos, actually slightly wrong? And a rather interesting counter-proposal came from Jim Lake a few years ago, where instead of having the tree of life, we have a ring of life. And this, to some extent, formalizes the fusion events and symbiosis that Lynn will talk about later on. So here we would have a situation where we have one lineage giving rise to archaea, one to bacteria, and then the eukaryotes actually arose via full fusion of probably a proteobacterium with, and in Lake's proposal, a Krenarchaeal ancestor. And my lab love this. Well, I love it, and I think my lab does. Because this means that the Krenarchaea are, in essence, a nice living fossil of the last common ancestor of archaea and eukaryotes. And we would argue that after the divergence of the Euryarchaea, we evolved all these complex features that are preserved in present-day Krenarchaea and, of course, continued and elaborated upon in eukaryotes. Okay, and the last, have I still got 2 more minutes?
Okay, yes.
2 more, okay, good, good. So the other question, specific question is, I'd like to address is, how did 2 distinct DNA replication machineries evolve? As I say, there's nothing more fundamental to the propagation of life on the planet than the ability to replicate your DNA. And yet we have 2 entirely distinct apparati that have emerged during evolution. There's a rather attractive proposals come from Patrick Fauter, who's based at the Institut Pasteur in Paris. And this is of a cataclysmic occurrence early in the evolution of life on Earth, not the planet-busting asteroid as I cartoon here, but on a slightly smaller scale, the collision of a virus with a host genome. So if we invoke a situation where we have a host organism with a single origin of replication, dependent on this single site for replicating its genome, and a virus happens to integrate into that host genome at this site, well then the host has a problem because you've taken out its origin of replication. It simply can't replicate anymore. Rather, the host now becomes dependent on the virus and the virus's own sequences and proteins to drive replication of the entire host chromosome. So if we invoke a time back in evolutionary history when the evolutionary tree of life was the evolutionary stump of life, we have a virus come flying in at the point of Luca, does its little replicon transfer, take over, and now it effectively hardwires this entire lineage with its own DNA replication apparatus, thereby supplanting the host machinery and creating this novel lineage, this novel machinery for DNA replication. I love the idea. I think it's extremely sweet. And there is, fortunately, some evidence for it as well. Mitochondrial DNA, and again, Lynn is one of the world's experts in this, the replication and transcription machineries are actually clearly derived from a phage. They're not classic bacteria, although the mitochondria itself is derived from a bacterium. It's actually a phage that's gone in and supplanted the bacterial machinery with its own machinery in order to drive mitochondrial replication. We've also discovered a bacterium, Bacillus cereus, which has an integrated phage within its own chromosome, which actually has archaeal and eukaryotic replication proteins, showing that we can get transfer between the domains of life. And also more recently, we've also shown that the archaeal genomes have integrated viruses within them that are actually helping to drive the replication by defining start sites of replication within the chromosome. So in conclusion, I'd like to propose, and something we can discuss later perhaps, that viruses not only shape the genomic content of host organisms, but may also play an important role in actually sculpting the architecture of replication and indeed the very machinery that's carrying out replication itself. And I'll stop there.
Thank you very much for keeping to time too with an exciting presentation. We move on to Martin Brazier, author of Darwin's Lost World.
Yes, if there's anybody who hasn't got this book, it came, which seems unlikely. It came out on Darwin's birthday. And my point in the book was really to emphasize a number of things. And one of them was that Darwin had, to some extent, misrepresented what the fossil record can tell us. And it has some very important things to tell us about evolution and deep time, particularly for the Precambrian, which is 80% of Earth history or thereabouts. Darwin had racked his brains as to how to explain the lack of fossils. And during my lifetime, there's been enormous progress on that. And I think paleontology shows that all the really important things that have ever taken place in evolution, the irreversible transformations, like the origin of oxygenic photosynthesis, actually take place in that great dark age, which is the Precambrian. But I don't want to talk about that here. Lynn has taken great interest in a course I give at Oxford to the third years, which really explores the uses of a particular fossil, which is the most used fossil in all of geology. And most biologists hear rather little about them. But for geologists, they're the fruit fly of our subject. We use them for stratigraphy, for environmental change, for predicting climate change, everything like that. So it's a huge field, and that's the study of foraminifera. And generally speaking, there are 5,000 or 6,000 foram specialists in the world. It's a big field, run mostly in oil companies and geological surveys. Now, like Darwin, I was lucky enough to spend a year as a ship's scientist or a ship's naturalist on HMS Fawn in the Caribbean, and my job was to actually look at the foraminifera as proxies to the study of deep time. And I don't cover it in this book, but I'm hoping to get to it in a later one. That's my name on the left. Jonathan at the Antcliffe is on the right there, and John's helping me to explore some ideas we've had over the last 10 years or so, just to illustrate foraminifera. Here's a slightly microscopic view of some modern-day forams, sort of thing you might find living in Hawaii or in the Caribbean today. And there's 3 forams, well, there's 4 or 5 you can actually see there. The one in the middle is a great big thing about 2 millimeters across. It's a single protozoan, a rhizopod protozoan called Heterostygina, and it lives almost entirely dependent on its symbionts, which are diatoms, a particular species of Nietzsche or something like that. And that's what gives it its golden colour. And down at the bottom there you can see one that's got red endosymbiotic algae inside it, and another one over on the left here which has got brown dinoflagellates. And biologists and geologists have discovered that there's a whole host of foraminifera which have specially adapted to cultivating their symbionts in just the same way as the famous reef-building corals do today. But the wonderful thing about the forams is they have the best fossil record of almost any group. It goes right back to the Cambrian, 540 million years ago. It's enormously diverse, and it can be read in small chips of rock or in thin sections, so it's a story we probably know better than any other group. One of the things, of course, that geologists like to do is to go to tropical lagoons like this and study reefs and forams. And there's a little chart you don't need to read the details of, but it shows the kinds of symbiont that different foraminifera have as you go into deeper water from the back reef into the forereef. And as you might expect, the shallow ones have adapted to cultivate chlorophyceae, that's green algae, adapted to the shallow water light levels. And then there are groups that have adapted to cultivate dinoflagellates and red algae, and they usually live at intermediate depths. And then there are forams that are capable of living about 120 to 150 meters deep, where the photic zone is very weak and feeble, but diatoms are specially adapted to low light levels. That's why they thrive in the high and low latitudes today. And rather marvellously, it turns out that the form of the skeleton reflects the nature of the symbionts that they cultivate. So we can actually use this to some extent. Foraminifera that are dependent on symbionts also change their shape and their wall structure according to the depth at which they live. So shallow ones have thick, robust tests, and as they have to live deeper and deeper in the water, where the light levels are dimmer and the light is normally bluer, they get thinner and flatter. And there are these, and there are a whole host of other tests we can use to reconstruct symbiotic foraminifera in the past. And this is something I've always been trying to get at. This is some mathematical modelling I did years ago to show that symbiont cultivators typically have very short lines of communication. They're very concerned about energy pathways and energy conservation. And this is what some of the fossil forams look like. Here's one, it's a bit bigger than natural size at the right there. It's normally a couple of centimetres across. This particular genus, Nummulites, gets to 150 millimetres across, 15 centimetres across, at the core of the Eocene 45 million years ago. It makes up the Sphinx and the pyramids today and is massively rock forming. And then there's another type in the late Permian, which again reaches this enormous size, called Fusulina here, also rock forming. And we know very much in detail the evolutionary history of these foraminifera as they become more and more specialized, apparently, to cultivate their symbionts, creating tiny little compartments in which the light can get through. Some even have fiber optic type devices of calcite to concentrate the light at greater water depths. And we've been plotting the architectural evolution of foraminifera, which starts very simple at the top there, and as you come down, you're going towards more and more of advanced architecture, so you get multiple chambers, and then you get lots and lots of openings between the chambers to let the symbionts in and out. And then in the ones that are really symbiont specialized, they create these little chamberlets, like little cubby holes, for the particular farming and the nurturing of the symbionts. So we know there's a whole series of indicators we can look at. And if we look back through geological time, and geologists use these for dating the rocks in the Permian and in the Cretaceous and so on, so we know the fossil record of these remarkably well, we can see there are cycles, major cycles. I tried plotting them out this way first, and the parsimony index means very simple and not symbiont cultivating, and number 10 at the right means probably symbiont cultivating. And here's geological time at the left here. So you can see in the Carboniferous and Permian there's a great radiation of symbiont cultivators, and then there's an episodic series followed usually by extinctions. And this plots it out in a bit more detail. There are typically three shapes that the symbiont cultivators like: spindle-shaped at the top right, Chinaman's hat-shaped at the top left, and coins-shaped down at the bottom. And we can see the evolutionary trend towards symbiont cultivators by the arrows. It shows the point at which symbiogenesis started to kick in, and the red lines are extinction, and they usually mean the total extinction of the lineage. So some of these are the very famous extinctions, like the one at the end of the Permian and the one at the end of the Cretaceous. In fact, all the big mass extinctions cause enormous collapse of the symbiont cultivating forams. And just to show you how that really works in terms of size, this is 140 microns at the right here. So you can see that there are cycles of maximum size taking place. And rather fascinatingly, all the big extinctions are preceded by very giant foraminifera. Now that is either an enormous coincidence, or it indicates to me that the ecosystem in which gigantism develops had something about the nature of its connectedness, and the collapse is related to some extent to the nature of the ecosystem and its vulnerability. So that's what I want to end with, really, to emphasize too that we can cross-check to some extent what happens to symbiosis, and that foraminifera and their symbionts have actually got their act together episodically. And as far as we can see, it tends to dissolve about every 20 to 30 million years or so. There are very few examples where it seems to have sustained longer than that. Okay, thanks.
Thank you very much, Martin. And so we ask Richard to give his few minutes of introductory remarks. Over to you, Richard.
Well, I thought I would talk about what's called neo-Darwinism, which sometimes comes in for some stick. And in particular, what can be called the gene's eye view, which is one particular way of looking at neo-Darwinism. The gene's eye view focuses on the level of genes, not specifically that they have to be DNA, although on this planet they are, but anywhere in the universe where there is self-replicating, coded information which can exert some power, which we call phenotypic power, some power over the likelihood that it will be copied on into the future. So the key point about the gene, which is a somewhat abstract concept rather than having to be specifically DNA, for the reason I've just said, the key point about the gene in this model, in the gene's eye view of evolution, is that it is potentially immortal in the form of copies, because the copying of genes, I'll call them that, is exceedingly accurate, not totally accurate, but very, very accurate. there is the potential possibility that the information will last forever. And that opens up the possibility of competitive struggle between rival versions of these replicating pieces of coded information. Because although they potentially can last forever, not all of them do, and those that don't are selected away by natural selection. So that is natural selection. Natural selection is the differential survival of coded information which exerts power over its probability, over its likelihood, over its success in being copied, in being replicated. And the power that it exerts is phenotypic power, which means it exerts power over embryological processes, the building of bodies usually, although I would rather generalize that and say any kind of influence that a self-replicating entity can exert over the world, which improves its probability of replicating itself and passing itself on to the potentially eternal future. Any kind of influence that it can have will be favored by natural selection. The unsuccessful ones perish, the successful ones don't. The vehicle of their success or failure is phenotypes. It happens to be the case that in the forms of life that I'm most familiar with, which is animals, these phenotypes are great big vehicles. They're great big multicellular entities that move around as a unit, on legs or wings, or fins or tails. They are things that have eyes and brains and kidneys and hearts. That is a very special and in a way peculiar way of doing it. It didn't have to be like that. That happens to be the way it is. The more general way of looking at the gene's eye view of natural selection is to say that any kind of phenotypic power that can be exerted will be. Now, the gene's eye view has been criticized as being determinist, as though we are saying that genes exert a deterministic influence on bodies which is irreversible and inevitable and simplistic. And I want to really repudiate that most vigorously. I yield to no one in my admiration of the complexity of feedback loops, of the details, the immensely complicated details whereby genes actually do influence phenotypes. There's absolutely no suggestion that it's irrevocably deterministic. There's absolutely no suggestion that it's simple. But however complicated it is, the one thing that matters is that individual differences between genes are reflected in individual differences in phenotypes, and it is those individual differences in phenotypes that count as far as selection is concerned. So it doesn't matter how complicated the influence of genes on phenotypes is. It doesn't matter how complicated the feedback loops are. It doesn't matter how much involved they are with symbiotic ingestion of other types of organism. This can happen to your heart's content. All that matters is that differences between genes, differences between self-replicating coded pieces of information are reflected at the end of this amazingly complicated piece of embryological development. Differences between coded pieces of information are reflected in differences in survival of those coded pieces of information. And that's the important point. That's the only thing that singles out the gene in the hierarchy of life as being important in the gene's eye view. It's not that they exert a more important causal influence on bodies. The only thing that matters is that they are potentially immortal, and differences between individuals are reflected in differences in survival via phenotypic differences, which can be as complicated as you like, and which can indeed involve symbiotic relationships with other creatures. And here I come on, obviously, to Lynn's particular interest. Lynn, of course, is the great apostle of symbiogenesis. I don't think she goes far enough. What I want to say is that things like the incorporation of bacteria to be mitochondria and chloroplasts and possibly other things, they're just the tip of the iceberg. We can regard the entire gene pool of any species as a symbiotic collection of self-replicating coded pieces of information. The other genes in the gene pool should be regarded as part of the ecological environment of every gene in the gene pool. So every gene in the gene pool is surviving against a background of not only the external environment, not only the weather, the humidity, the rainfall, the predators, the parasites, the hosts, not only all of that, but also the other genes in the gene pool. And so the gene pool of, say, the lion species is one particular collection of symbiotic genes who are all cooperating with one another, with other members of the gene pool to make lions. And similarly, the buffalo gene pool and the kangaroo gene pool, they're all symbiotic collections of genes in just the same kind of way as Lynn has taught us with respect to symbiotic organisms. So in that sense, I think she hasn't gone far enough.
Thank you very much, Richard. Yes, I'm actually the moderator and chairman of this, but of course I can't hide the fact that I'm also a physiologist who's interested in heartbeats, in how brains work, and so on. And so a lot of my work focuses on the question of how collections of genes, or rather collections of gene products, interact to produce the complicated function that we observe as physiologists, whether it be the heartbeat, whether it be the secretion of insulin by the pancreas. All of these functions are functions that physiologists study by looking at the way in which the gene products interact. I will just say one thing about that interaction, which I'd like you to take into account as we proceed in this discussion. How big a space could that interaction be? If you calculate the total number of interactions there can be between 25,000 genes, which is the present number of genes identified in the human genome, it comes out to be 10 to the 70,000. I'm rounding the figures. You can imagine an exclamation mark after that, and the exclamation mark, of course, would not be an indication that that is factorial. How many, I've tried once to see, how could one compare that number with a number which we can, as it were, grab hold of? And I thought the best number to compare it with, remember 10 to the 70,000, I thought the best number to compare it with would be the total number of atoms in the universe. As Hubble has probed into deep space and revealed those tens of thousands of galaxies, even in just 1/1,000,000th of the space of the sky, how many do you think we end up with? 10 to the 80. There wouldn't therefore be enough atoms in the whole universe for evolution to have, as it were, serendipitously tried out all of those interactions, even over the billions of years of life on Earth. And there's my contribution as a physiologist to this. We therefore have to work out how evolution has serendipitously done it, how through the various paths it's chosen, which I would suggest is only one path out of billions of paths, and if we do find life on some other planet sometime, maybe it'll be very, very different from us. I'd love to hear the reactions of my colleagues on that point. To a physiologist, at least, as I said, the number of possible interactions that we're looking at is absolutely immense. Well, there's my sort of little contribution to the debate. I think at this point, then, we open up for people from the floor to make comments or questions. Please stand up, say who you are.
I'm a developmental biologist.
Please.
I want to take issue with Professor Dawkins on the grounds that it's just not terribly helpful to take a gene's eye view. It may not be wrong, but it isn't much use because selection doesn't act on genes, it acts on phenotypes too complicated. We know. I mean, one of the most distressing experiments anybody can do is to knock out a gene and see that it has not even a subtle phenotypic effect. We know that the individual genes are the jigsaw puzzle pieces. But you do need sort of images to make sense of things. And you do need real phenotype for selection. So I just don't think it's terribly helpful.
Well,
Do you want to react immediately to that?
I mean, I think.
I won't do this for every comment and question, but I think Richard
Of course, we don't disagree about that. You do need to actually look at phenotypes. However, when you do look at phenotypes, and when you ask the question as a field biologist does, what is the adaptive advantage of a certain piece of behaviour or a certain or a certain phenotype, you do need to ask at what level you're asking that question. And field biologists nowadays almost all take it to the level of the gene. They're looking at phenotypes, they're looking at tails and wings and behavior patterns and everything else. But the question they're actually asking is, how is it that this behavior pattern or this morphological feature increases the survival chances of genes that don't make it, because obviously, as you say, a single gene doesn't make a phenotypic feature, but make the difference between an individual that has this variety of phenotype and one that has that variety of phenotype. And so the rhetorical convention, what's in it for the gene that makes this thing, is the way people actually work. It's enormously fruitful. If you go out to the Serengeti, if you go to the Antarctic, wherever field biologists are now working on adaptive questions, that's the question they're asking.
Next point, yes, please, say who you are.
Robert Siegel, and I want to address this to the first speaker. As a virologist, my heart beat faster to hear your theory of the role of the virus, but doesn't that really just push the question backwards, because you have to actually explain where the virus got it, and presumably it's likely that there's only one origin for the enzyme in history, so you just actually pushed it back in time.
So one answer, I guess, is that the number of generations that viruses go through will scramble the coding sequence, will increase the chance of diversity occurring. What we also need to recall, though, is that we're currently looking at 3 domains of life. Those are the ones that are still existing on the planet today. There are probably 2 billion years prior to the divergence at Luca, where other lineages have emerged, they've tried various combinations out, they've maybe become extinct. It's formally possible that viruses may actually reflect ancient fossils of those ancestral, now extinct lineages. And so the diversity, again, we're just pushing the question back further.
Yeah, so the real question is, do you think that it evolved independently more than once, or do you just spread it out in time so that you could get more diversity?
What we can do is take it back to the point where the genome itself wasn't DNA, but rather was RNA, in which case we then have a takeover point where possibly distinct lineages evolve different mechanisms for generating DNA and replicating it. Why hasn't that simply happened in Luca? Why hasn't Luca got an RNA genome? Formally possible that it could, but we note that bacteria, archaea, and eukaryotes all share a common mechanism for repairing DNA. The homologous recombination machinery is the same in those 3 lineages, suggesting that that was present in Luca, which in turn suggests that Luca was repairing DNA genomes, therefore Luca itself had a DNA genome. So the RNA-based genome, we would say, was earlier in time.
Steve, please say what Luca is.
Sorry, the last universal common ancestor.
Eric, you wanted to say something.
Eric Werner. Yeah, I have a question to Professor Dawkins about the symbiont idea of genes. And one of the problems that I see with that is, in effect, are you saying that they somehow interact to then come up with the organism? Because, I mean, in effect, I'm wondering whether there's sufficient complexity just in the pure interaction between genes as such versus the networks that actually construct genomes. So I don't know if you can view her view as I don't know if you can really take genes to be symbionts, because that would in effect make them cooperative entities that then through their interaction produce some complex structure. And that has inherent limitations because of the relative simplicity of those entities. So you need a temporal cascade of some kind, which isn't necessarily in the proteins.
Do you want to comment on that now or to hold it for later?
Let's hold it for later.
Hold it for later, is that okay? We'll hold a number of things like this. Yes, please. And say who you are.
My name is Michael Isaac. My training, such as it was in history, but I've done some other things. I'm going to ask Professor Dawkins and everybody else present. As far as I understand it, there's a huge volume of mass of genetic material which hasn't been selected out. It's called non-recording or A-recording DNA. And I'm not asking for an answer, and I wouldn't dream of asking you, Professor Dawkins, whom I might disagree on other matters, and that is why, that is, can we give an account of that?
Yeah, okay. So the non-recorded...
I'm sure you'd rather leave it.
No, I think it'd be good to put this to any of the members of the panel.
I don't need to
Non-recorded
answer.
DNA.
I find it very difficult to put it forward in such a crude way.
Yeah.
I can't believe that this huge mass
Is there doing nothing?
it is standing there doing. I'm quite sure it isn't.
I am
There
quite sure
are
she's
very
not
few
doing nothing.
witnesses who can consider this.
I think it might well be doing nothing.
Well, okay. Richard first. Do you think it may well be doing nothing?
In that case, Professor Dawkins, all I can say in that particular case, so you, because it's got to be explained.
Well,
Richard, do you want to come in first?
It's very easy on the gene's eye view to explain it because it is simply the ultimate parasite, as Crick and Orgel said. I mean, it's got the perfect way of replicating itself. It doesn't affect anything, so it's not selected against.
I don't think that's
Let me make another suggestion. Please, yeah, we've got the question, we've got the comment, and we're going to now try and reply. I'm going to give a slightly different reply from Richard, but we'll see also what the other panelists want to say, because I think the... I don't want to say anything. Well, consider this. When, as a physiologist, I look at the way in which a protein functions, of course I have to focus on the active sites of the protein, the bits that really make it be the receptor for this, the channel for that, or whatever its function is. But we have a very similar problem, you know, with proteins, because there's much else which contributes eventually to the three-dimensional structure of the protein. And the way we solve the problem there is that we say, the problem you're posing, what on earth is it doing, is we say, well, that takes part in forming the three-dimensional structure within which the sites are to be found. And that has a very big effect on the speeds of interactions, and therefore it does have a function. Now, my suggestion would be simply that the genome has to be folded up. We know, of course, that the DNA is wound around histones and all the protein machinery that the nucleus has. We know also that many genes, particularly in the higher animals, are broken up into many introns and exons. My suggestion is that when we look at the way in which the whole thing is folded, and which exon comes close to which other exon, we may be able to start to say why it is that particular splice variants appear rather than others, or in higher frequency than others. So I don't think it's too difficult using the analogy with proteins to give some sort of functionality, particularly in the folding mechanisms that may determine which genes are expressed and to what extent they're expressed. Now, this is just an idea. I don't think anybody can prove this. But over to others. Richard, do you want to come in first on that?
There are related species of salamander which have orders of magnitude difference in genome size. And that doesn't... I mean, any kind of functional explanation is pretty improbable. It's far more likely that this is just rubbish that's left lying around on the hard disk and just hasn't been cleaned off.
So it would be fossilized in effect.
Well,
These are basically fossil genomes.
Our instincts obviously disagree on this, but let's...
Well, how would you explain the salamanders then?
I don't work on the salamander, Richard. What I do work on is the fact that taking, for example, DSCAM, one of the genes in the fruit fly, that has something like 35,000 different ways in which its own spread of exons can be expressed. And what we know is that those are expressed at different times during the development of Drosophila. So something is interacting to determine that. Whether it is the junk, as we call it, DNA, that is helping to form the structure or not, I can't say. I'm only putting an idea out. We have to explain that, though, because it's clear that something else is determining the order in which the expressions are occurring, in addition to what is in the DNA sequence in the exons themselves. That's, I think, the essential point that I would want to make. But maybe we leave this also on the table to take up a little bit later, and I'll make a note of it. Stephen, do you want to say anything on this issue?
I just say that I agree with you that a lot of the DNA probably plays an architectural role, but we also have to recall that until recently we didn't know about the existence of small non-coding RNAs that are actually involved in the control of a great number of genes. That was previously thought to be DNA that wasn't having any coding function. Now we know it actually plays pivotal roles in regulation. So a lot of DNA, I think, will have as yet undiscovered roles the sort of microRNA type families of genes being one example.
Who else wants to come burning in? Right at the back there. Again, can you say your name and then put the point you want to make?
My name is William Nash. I'm an undergraduate geologist. And Professor Braiser, you mentioned that following the PT and KT extinctions, there was gigantism in the foraminifera. Does anybody know of any possible explanations for that? And if so, could we use those explanations maybe to explain the causes of the extinctions? Because they're, I believe, certainly in the case of the KT extinction, rather controversial at the moment.
Yes, we've done isotope work, and it looks like they lived quite a long time. So some of these foraminifera lived tens to possibly as much as 100 years. They were probably optimizing their moments of reproduction. That's just what I'm guessing anyway. And probably living fairly low in the low down in the photic zone, waiting for the right moment in a way that corals do. At least they reproduce about once a year or thereabouts. As you know, mass extinctions tend to hit very large things with rather slow rates of reproduction and recruitment, so it probably explains the sorts of things that are going on. More than that, I wouldn't want to interject at this point, I think. I'm not sure it throws too much light on the discussion.
Okay, we take a 10-minute break while the film gets set up.
Exactly.
Is that
The lights
correct?
get turned on.
And then
Yeah.
you'll say something to introduce
Yes.
it, Lynn. Okay, a 10-minute break to let people get some fresh air.